Welcome back to the Biology Bros with Luch, Gavin Brown, George Watson, and Mr. Chisholm. Today we're going to talk about genes and heredity. Let's get into it.
First off, quick summary of 12.1. You can't talk about genes without talking to first mentioning Johann Gregor Mendel, the father of genetics. He spent years of his life crossbreeding a massive amount of pea plants, 30,000 to be exact. Mendel wasn't just randomly growing plants, he was systematically tracking traits like plant height, seed shape, flower color, and pod characteristics. When Mendel crossed plants with different traits, he discovered something fascinating in the first generation. All offspring showed only one parent's trait, the dominant trait. For example, if a tall plant was crossed with a short plant, all F1 offspring would be tall. But here's where it gets interesting. When these F1 plants were allowed to self-pollinate, the F2 generation showed a predictable pattern. Approximately 3/4 of the plants displayed the dominant trait, while a staggering 1/4 displayed the recessive trait. This 3-to-1 ratio was discovered and it was revolutionary. He proved that traits are passed down through discrete units we now call genes, and that some traits can be hidden but still transmitted through future generations. His meticulous research transformed our understanding of how characteristics are inherited, setting the stage for modern genetics and our comprehension of heredity.
Now, we will begin to go more in depth. First, furthermore with Mendel, he was a learner, teacher, scientist, and a man of faith. When he was a young adult, he joined a monastery called the Augustinian Abbey of St. Thomas in what we now know as Czech Republic. There, he taught physics, botany, and other natural science courses at multiple levels. In 1856, he started research about the inheritance patterns between honeybees and plants. In the end, he decided his test subject should be pea plants as his primary model system. Later, in 1865, he experimented with about 30,000 pea plants, demonstrating that traits are transmitted from parent to offspring in recessive and dominant patterns. At first, his work was unrecognized by scientists who at the time believed that the process of inheritance was a mixture of traits from each parent, which we now know is only true in some circumstances. This mixture of traits is called continuous variation. An example would be eye color. Instead of this, Mendel researched and experimented with what's called discontinuous variation, which is where traits are inherited in distinct classes. He ended his studies in 1868 and switched completely to his pastoral duties. His work was rediscovered, reproduced, and brought back to life in 1900 when scientists came across it once more. Furthermore, with his pea pod experiment, he used the garden pea, which self-fertilizes when pollen finds the ova, blooming, creating true breeding or inbred plants. These plants always look the exact same as the parent. Mendel used this true breeding plant to eliminate the chance of other unexpected traits. He also used large quantities to conclude that his results didn't just come by chance. He also used hybridizations in his experiments, mating 2 true breeding plants with different traits. This resulted in the discovery of P0, F1, and F2 generations, and the public start of recessive and dominant traits.
Welcome back, guys. Now we're going to talk about recessive and dominant traits. First generation cross plants are in the P0 or parental generation. Mendel then collected the seeds from this generation and grew them the next season. This generation of seeds is called the F1 generation. In his experiment, Mendel let them all self-fertilize and he collected the seeds from this plant. Finally, he planted seeds from this generation to create the F2 generation. His experiment extended past the F2 generation, but the results from the P0, F1, and F2 generations were the most intriguing. For this experiment, Mendel used violet and white flowers. He applied the pollen from the violet flowers to the stigma of the white flowers, creating the next generation, F1. He found that this generation was entirely violet flowers. This proved that the white flowers from the P0 generation were completely gone for now. He then allowed the F1 generation to self-fertilize, which resulted in 705 violet flowers and 224 white flowers. This ratio of violet to white flowers was about 3 to 1. Even when he applied the pollen from the white flowers to the stigma of the violet flowers, the results were the same. This showed a skipped trait from generation to generation, aka a recessive gene. The purple trait that was shown the entire way through is known as a dominant trait.
Now for the last part of 12.1, equations. Mendel determined that traits like seed color and flower color are passed down independently from each other. Performing a crossbreed with a pea plant with green and wrinkled seeds and a plant with yellow smooth seeds, Mendel found out that the round seeds produced offspring that had a 3 to 1 ratio of yellow-green seeds and a 3 to 1 ratio of wrinkled to round seeds. A thing called product rule can be applied to Mendel's findings of independent characteristics. Product rule is the probability of two independent events occurring together. Can be calculated by multiplying the individual probabilities of each event occurring alone. To explain this, imagine you roll a d6 and a penny. The d6 can land anywhere from 1 to 6, and a penny has heads or tails. The rolling of the dice and the flipping of the penny have no influence on each other, resulting in 12 possible outcomes. A die roll has a chance of rolling a 2, and a coin flip has a chance of landing heads. The probability of rolling a 2 and flipping heads is 1 in 12 chance. On the opposite side is the sum rule, which is a probability principle used to calculate the likelihood of either one of two mutually exclusive events occurring. It states the total probability is the sum of their individual probabilities. For example, in heterozygous cross, the probability of offspring having a dominant phenotype is calculated by summing the probabilities of each genotype. This rule is essential for predicting phenotype outcomes in Mendelian inheritance.
When true breeding, homozygous individuals differing in a trait are crossed, their F1 offspring are heterozygous and exhibit the dominant phenotype if the trait follows dominant recessive inheritance. Self-crossing these heterozygotes produces F2 offspring and a genotypic ratio of 1 to 2 to 1, homozygous dominant to homozygous recessive, and a phenotypic ratio of 3 to 1, dominant to recessive. However, not all traits follow simple dominance patterns. One is incomplete dominance, which is when the heterozygote has an intermediate phenotype. Another one is codominance, which is when both alleles are expressed simultaneously in the heterozygote. Finally, multiple alleles, which is when more than two alleles for a gene can exist in population, though individuals only inherit two.
Now, the last part of 12.2. Human females have a homologous pair of X chromosomes, whereas human males have an XY chromosome pair. Although the Y chromosome contains a small region of similarity to the X chromosome so that they can pair during meiosis, the Y chromosome is much shorter and contains many fewer genes. In fact, when Nettie Stevens discovered that the X and Y chromosomes were the determinant of sex, she differentiated them only by size.
Now on to recessive lethal alleles. Homozygous individuals die while heterozygous survive, with a 2 to 1 ratio in offspring. Dominant lethal alleles, fatal in both homozygous and heterozygous, but can persist if lethality occurs post-reproduction, as seen in Huntington's disease.
Next, here's a summary of 12.2. When true breeding or homozygous individuals that differ for a certain trait are crossed, all the offspring will be heterozygotes for that trait. If the traits are inherited as dominant or recessive, the F1 offspring will all exhibit the same phenotype as the parent homozygous for the dominant trait. If these heterozygous offspring are self-crossed, the resulting F2 offspring will be equally likely to inherit gametes carrying the dominant or recessive trait, giving rise to offspring of which 1/4 are recessive. Because homozygous dominant and heterozygous individuals are phenotypically identical, The observed traits in the F2 offspring will exhibit a ratio of 3 dominant to 1 recessive. Alleles do not always behave in dominant and recessive patterns. Incomplete dominance describes situations in which the heterozygote exhibits a phenotype that is intermediate between the homozygous phenotypes. Codominance describes the simultaneous expression of both of the alleles in the heterozygote. Although diploid organisms can only have 2 alleles for any given gene, it is common for more than 2 alleles of a gene to exist in a population. In humans, as in many animals and some plants, females have 2 X chromosomes and males have 1 X and 1 Y chromosome. Genes that are present on the X but not the Y chromosomes are said to be X-linked, such that males only inherit 1 allele for the gene and females inherit 2. Finally, some alleles can be lethal. Recessive lethal alleles are only lethal in homozygotes, but dominant lethal alleles are fatal in heterozygotes as well.
We now move on to 12.3, the laws of inheritance. Mendel proposed that hereditary traits are governed by paired unit factors, genes, that segregate and reassociate during gametogenisis and fertilization. His pea plant experiments revealed that recessive traits hidden in the F1 generation reappeared in the F2 generation in a 3-to-1 ratio. This led him to deduce that traits are inherited as discrete units, contradicting the prevailing belief in trait blending.
Mendel's law of segregation states that paired genes, alleles, separate equally into gametes, giving offspring an equal chance of inheriting either allele. In a monohybrid cross, the F1 generation shows only the dominant trait, while the F2 generation exhibits a 3-to-1 phenotypic ratio due to 3 genotype combinations. 1 homozygous dominant, 2 heterozygous, and 3 is homozygous recessive. Heterozygotes arise from 2 pathways and are phenotypically identical to homozygous dominance. This law underpins the use of Punnett squares for predicting offspring genotypes. Its physical basis lies in meiosis 1, where homologous
Mendel's law of dominance states that in heterozygotes, the dominant allele masks the recessive allele, which remains latent but can be passed to offspring. The recessive trait is expressed only in individuals with 2 recessive alleles. However, later research revealed that the law of dominance is not universal, as other inheritance patterns also exist.
Mendel's law of independent assortment states that genes are sorted into gametes independently, allowing all allele combinations to occur with equal likelihood. This is demonstrated in dihybrid crosses such as between YYRR, yellow round, and YYRR, lowercase, green wrinkled pea plants. The F1 generation is uniformly capital Y, lowercase y, capital R, lowercase r, and self-crossing these heterozygotes produces F2 offspring with a 9-to-3 to 3-to-1 phenotypic ratio, round/yellow, round/green, wrinkled/yellow, wrinkled/green. This ratio reflects 2 independent 3-to-1 monohybrid ratios for seed color and texture. Independent assortment occurs due to the random alignment of homologous chromosomes during meiosis.
Mendel proposed that genes, which determine traits, are inherited as pairs of alleles that follow a dominant and recessive pattern. During gamete formation, the 2 alleles in a diploid organism separate, ensuring each gamete has an equal chance of receiving either allele. Additionally, genes are distributed into gametes independently of one another. This means that an allele for one gene does not influence the segregation of an allele for a different gene. Independent assortment is demonstrated by dihybrid crosses when the genes being studied are located on different chromosomes or are far apart on the same chromosome. For genetic crosses involving more than 2 genes, it is more practical to use methods like the forked line approach or probability calculations rather than the Punnett squares
predict offspring genotypes and phenotypes. While chromosomes themselves sort independently during mitosis, Mendel's law of independent assortment applies specifically to genes, not chromosomes, which carry thousands of genes. When genes are located close together in the same chromosome of their alleles, they tend to be inherited together, leading to the offspring ratios that deviate from Mendel's predictions. Recombination helps shuffle genetic parental alleles to mix on the same chromosome. This is why alleles on the same chromosomes are not always inherited together. Recombination occurs randomly along chromosomes, so genes that are far apart on the same chromosome often still assort independently due to recombination events in the inverting space. In some cases, genes can interact at the level of their products. This interaction may result in one gene's allele masking or modifying the expression of another gene's allele, a phenomenon known as episteinsis.
That's it for the Biology Bros today. Catch you next time. This was Luch, Mr. Gatt, George, and Ian as your writer and editor. Catch you later.